So I'm actually coming to you from sunny Turkey today, funnily enough, not Greece, but we're close. So I've been looking at alternative splicing in cell differentiation with a particular focus on what long read sequencing can enable us to discover more. Next. So alternative splicing is a really important mechanism that happens to pre-messenger RNA to make mature messenger RNA, and it essentially allows us to chop and change exons into differing arrangements. Next. So you can see a multiple variety of mechanisms here, and essentially the main gist is to put these exons into new arrangements, which gives us differing transcripts and differing proteins. Next. This is really important in things like neuronal processes because these can be regulated by alternative splicing, and also in organs like the brain, we've got a huge need for ongoing neuroplasticity. Next. We also really care about splicing because when it goes wrong, many diseases can arise. So we want to find out more about what's going on. Next slide. So I've been using long read sequencing to make a bit more sense of this. So at the moment, most of the transcriptomic and genomic annotations have all been made with short read sequencing. This is really great, but however, we're missing out on a lot of things. Next. So the group that I've been working in before I came along, they used long read sequencing on this one single gene, CACNA1C, and they were able to find 38 novel exons. So if we're caring about what's happening with these exons, we want to be able to know the other exons that we can't even detect at the moment. Next. So I used in my analysis a custom annotation that was made using Oxford Nanopore technology long read data. And this new genomic annotation had all of these novel features next that you can see in the bottom right hand side. So I used a neuronal cell differentiation model to essentially look at all these long read new features and see what more can we find out about alternative splicing by applying long read technology. Next. So I applied an RNA-seq pipeline, pretty basic. We're just aligning reads. We used Illumina and Nanopore so we can orthogonally validate. And then I used a package called DXseq. DXseq essentially assesses something called differential exon usage. So I was looking at how exons were used before and after my treatment, which was differentiating the cells. Next. So as you can see here, we had a ton of genes that were showing this differential exon usage. So we know that alternative splicing is going on. Next. So you can see that we've got genes that are shared. We find them in both long read and short read data, but also we had a ton of genes that we found only in the long read data having this differential exon usage. Next. So I filtered these genes because there's a ton of them going on. I don't really care about all of them. So I filtered them and I looked at the ones that were upregulated in the differentiated cells, the ones that had these novel new cassette exons that were annotated using this Talon software, and ones that were expressed in brain tissue and related to neuropsychiatric disorders. So I'm just going to give you a few examples, well, a couple of examples of the really interesting ones that we found. Next. So. Firstly, which was really cool, there's a gene called TP53IMP2, and this was upregulated in our differentiated cells. However, it was only in the long read data that we could capture this differential exon usage. Next. So you can see that there's this really long 3' UTR region in the transcript, and this exon was actually upregulated in our differentiated cells, which actually made a non-coding RNA with a function. However, because this region was so long, over 3,000 nucleotides, the short read just couldn't even detect that this was happening. Next. And then also, next. So we had a gene called CACNA2D2, which is a psychiatric risk marker. It's part of calcium voltage-gated channels. And this is really interesting. So our long read annotation was able to annotate, next, this cassette exon, this cassette exon, which actually formed a novel transcript resulting in a truncated protein. And then furthermore, next, we had an area on the transcript that showed massive downregulation in our short read data, but without even understanding that this novel transcript existed, we wouldn't understand why on earth all these exons were being downregulated. So in summary, what I've essentially been able to show is that long read data can not only give us new annotations about the genome, but it also can help us make more sense of what's in our short read data. Thank you. Next slide, thanks. I'd like to thank the Genetic Society for the funding, the Hayati group at the Earlham Institute that I've been working with, the collaborators in the Tunbridge Lab at Oxford, and then here's a bunch of skills and fun stuff I've had a load of fun doing in this project. Thank you.